The last bacterial structures we'll talk about are the cytoskeleton, storage granules, and gas vesicles. The cytoskeleton is similar to the eukaryotic cytoskeleton, and it has similar functions as well. It functions in controlling the cell structure and controlling movement of materials within the cell, as well as cell division, which is similar to the eukaryotic cytoskeleton. But bacterial proteins differ from the eukaryotic proteins. And so they do have the cytoskeleton, they just don't have the same proteins that make up the cytoskeleton in eukaryotes. Bacteria also have storage compartments. These are called granules, and they allow for nutrients to be stored inside of their cells. They can also store waste products until they can get rid of them. Then gas vesicles are filled with gases like oxygen or carbon dioxide and allow the bacteria to move up and down in water so they can move towards nutrients or away from predators. Endospores are only formed in some bacterial cells. The two most common groups of endospore formers are Bacillus and Clostridium. We will be messing with Bacillus in our lab, and you'll actually get to do fun endospore activities with them. Endospores are cell structures that can persist in the environment when the environment is not favorable for life. So real dry or not enough nutrients, very hot, the endospores can persist. And when the environment becomes favorable, endospores will germinate. That's not the same as reproduction via binary fission, because we're producing the exact same cell that was there that formed the endospore. So endospore formation is a complex process. You start with a vegetative cell in the absence of nutrients or in unfavorable conditions that will start to undergo basic binary fission. So it replicates its DNA, but it forms a septum between the two structures of DNA, forming a smaller and a larger cell structure. The smaller cell structure becomes a forespore, and the parent cell actually wraps around the forespore. And so as the parent cell starts to degrade, the plasma membrane so you have the membrane around the forespore, and then you have the mother cell membrane circle it as well. You get layers of peptidoglycan that fill in between the two membranes, and then you add certain chemicals that help to dehydrate the cell structure. And so here's endospore formation.
Spore-forming bacteria grow as vegetative cells and divide by binary fission when nutrients are available and environmental conditions are not adverse. When nutrients are depleted or conditions become adverse, spore formation is initiated. The DNA condenses and aligns itself with the center of the cell. The vegetative cell is now referred to as the mother cell. Next,
Spore-forming bacteria grow as vegetative cells and divide by binary fission when nutrients are available and environmental conditions are not adverse. When nutrients are depleted or conditions become adverse, spore formation is initiated. The DNA condenses and aligns itself in the center of the cell. The vegetative cell is now referred to as the mother cell. Next, the DNA divides into two complete copies, and the mother cell membrane invaginates to form the developing forespore. The mother cell membrane continues to grow and engulfs the developing spore. The developing spore is now surrounded by two membrane layers.
So this table just shows the
Next,
different structures that we've
peptidoglycan is
talked
laid
about
down between the two membranes of
and the
developing
bacterial
spore
cell.
to form the
Now we're going to get
cortex.
into the
Dipicolinic acid is
eukaryotic
formed
cell.
inside the developing spore, and calcium enters from the
Eukaryotes
outside.
are much
As
larger.
calcium enters the
They
spore,
typically are
water
more
is removed.
complex. They can be single-celled or
A
multicellular.
protein coat
They
forms
have
exterior to the cortex,
organelles
and the
associated
spore
with
becomes
them.
mature. Some spores form an additional
And
layer called the exosporium.
down here you can
A
see
mature spore is resistant to
an
environmental conditions.
animal cell
Finally,
and a
lytic
plant
enzymes
cell.
destroy the mother cell, and the
So
mature
eukaryotes
spore
have
is
lots
released.
of stuff going on inside their cells compared to what prokaryotes have. Organelles are what allows us to function the way we do. The organelles are tiny little organs inside of the cell that have certain jobs to do. The plasma membrane of a eukaryotic organism is similar to that of the prokaryotic organism. There are some differences. So the plasma membrane is a double membrane with the phospholipid heads on the outside and inside of the cell, and then the tails in between, embedded in between. There are proteins within the plasma membrane, either integral proteins that can act as channel proteins or carrier proteins, and then we have other proteins that act as enzymatic proteins or as receptors or communicators proteins. Strengthening the plasma membrane, we have cholesterol in animal cells, and we have similar steroid molecules like ergostrol in the fungal cells. The energy production of eukaryotes occurs in an organelle called the mitochondria instead of in the plasma membrane like we see it in bacterial cells. They have that electron transport chain occurring inside of their plasma membrane. Ours occurs in our mitochondria. So I mentioned transport. Some of the cells, so within the plasma membrane, we have simple diffusion where molecules move from higher to lower concentration if they are small and nonpolar. We also have channels that allow for facilitative diffusion or active transport of movement of molecules against their concentration gradient. But we also have a function that is different from prokaryotic cells. Eukaryotic cells can undergo endocytosis or exocytosis. Endocytosis is taking in molecules, while exocytosis is releasing those same molecules. So with endocytosis, the plasma membrane forms around something external, the cell, and then pulls the material in, forming a tiny vesicle. With exocytosis, a vesicle that's inside the cell actually binds to the plasma membrane and then opens, releasing the contents outside of the cell. There are three forms of endocytosis that we typically talk about. Pinocytosis is the movement of tiny droplets into our cells. In humans, almost all of our cells can undergo pinocytosis. Phagocytosis is the movement of large molecules or large materials, so it's considered cellular eating, whereas pinocytosis, cellular drinking. These can only occur by certain eukaryotes, and in our bodies, in humans, some of our white blood cells can undergo phagocytosis. Receptor-mediated endocytosis is a specific form of endocytosis where only certain molecules are going to be phagocytized. And then exocytosis is the opposite, pushing materials back out. Ribosomes, I've talked about these before, but ribosomes in eukaryotes differ from the ribosomes in prokaryotes. Ribosomes in eukaryotes are called ADS ribosomes, and they're composed of a 60S large subunit and a 40S small subunit. And they function, though, in the same process, protein synthesis. The cytoskeleton forms the framework of the cell. It helps to maintain the integrity of the cell, movement of materials within the cell, as well as forming the mitotic spindle fibers that will attach to our DNA and pull our DNA when we are going through replication. The three proteins associated with the cytoskeleton are actin filaments, microtubules, and intermediate filaments. Flagella and cilia are similar to bacterial flagella. They are not composed of microtubules, though, and the flagella typically allow the bacteria to move in a straight motion, which is very different than the bacterial flagella, which cause them to move in a run and tumble.